everyone, and welcome to this webinar hosted by Jen and sponsored by Wuxi Aptech. Today's topic is Rare Disease Therapeutics, Accelerating Development and Improving Access. Thank you so much for joining us. I'm Uduak Thomas, Senior Editor with Jen, and it is my pleasure to be your host for today's webinar. We have three great speakers lined up for you. They are Dr. Huan Tan, an Associate Director at Wuxi Aptech, Dr. Alice Zhang, Director of Strategy Support at Wuxi Aptech, and Dr. Lin Liu, Vice President for Molecular Biology and Gene Therapy at M6P Therapeutics. We'll get to their presentations in a few moments, but first, let's go over some background. Advances in the understanding of rare disease biology, coupled with innovative technology and therapeutic platforms, has led to improvements in the research and development of drugs for rare diseases. Strategies such as enzyme replacement, cell therapy, and oligonucleotide therapeutics create a diverse toolbox for tailored interventions, leading to an increase in drug approvals. Despite this progress, many rare diseases still lack useful treatments, and the need for new approaches remains high. In this Gen webinar, our expert speakers will discuss important trends in rare disease therapeutics and highlight ways of accelerating the drug discovery and development process. The webinar will provide a comprehensive and stimulating perspective for the many research teams that are committed to increasing drug accessibility for people living with rare diseases. Today's presentation will be followed by a live Q&A session. If you have a question for our speakers, you can enter it at any time in the Ask a Question panel on your screen. All right, let's get started.
Thank you everyone for joining today's webinar. My name is Alice Zhang from Wuxi Aptech. We'll be joined by two expert speakers today. Firstly, Dr. Juan Tan, Associate Director from Wuxi Aptech, who is responsible for the rare disease platform, and Dr. Lin Liu, Vice President for Molecular Biology and Gene Therapy from M6P Therapeutics. In today's talk, we will discuss important trends in rare disease therapeutics and highlight ways of accelerating the drug discovery and development process. Recent advances in biology, technology, and therapeutic modality strategies have supported the continued growth and expansion of research and development for new treatments for rare diseases. Currently, there are approximately 7,500 therapeutic products with an indication for rare diseases globally, with very active preclinical and clinical pipelines. A wide range of therapeutic areas are being targeted, with cancer, neurology, and infectious disease being the top three. In recent years, we're seeing a rapid diversification of therapeutic modalities. Looking at the FDA approvals, there is an increasing prevalence of new modalities, especially gene therapy and nucleic acid therapies, although small molecule remains the most prevalent modality. The clinical development pipeline is showing a similar trend. If we look further into the modality breakdown, we can see that AAV viral vector gene therapy is the most prevalent modality for gene therapies, and for nucleic acid therapies, antisense and siRNA are the main modalities. The emergence and rapid growth of oligonucleotide and gene therapies, both ex vivo and in vivo, have brought many new treatments for rare diseases. There has been 27 approvals in these new therapeutic modalities globally in the past 8 years, and we expect more to follow. Despite this progress, many rare diseases still lack useful treatments, and the need for new approaches and ways to accelerate the drug discovery and development process remains high. I will now hand over to my colleague Juan to talk about preclinical drug development for rare diseases.
Thank you, Alice, for your opening and nice introduction. Good morning, good afternoon, and good evening, everyone. I'm Juan Tan from Wuxi Aptech, and I'm taking the lead of the rare disease platform at Wuxi Aptech. I'm excited to delve into the preclinical part of drug development for rare diseases. We know that rare diseases are disorders which affect a small number of people compared to the general population. The Orphan Drug Act defines a rare disease as a disease or condition that affects less than 200,000 people. But this definition that over 7,000 kinds of rare diseases affecting more than 300 million people worldwide, mostly children. So it is safe to say that rare diseases are not so rare, either from the perspective of disease types or affected population. With all those 7,000 kinds, around 72% are genetic disorders. Due to the limitation of awareness and specialized testing and the overlap with common diseases and the genetic complexity, it takes years to diagnose a rare disease on average. To date, only less than 10% of the rare diseases have effective treatment. So there is an unmet need to develop therapeutics for rare diseases. Just like for common diseases, the process of drug discovery and development for rare diseases typically include disease or target selection, modality selection, assay and platform and model development, and then hit and lead identification and optimization, and IND filing, all the way to clinical trials, and finally clinical use. Today we're going to focus on how we can do the selection of rare disease and targets and the application of different modalities in rare diseases, and how in vitro and in vivo models can facilitate the process of drug discovery. Firstly, for disease and target selection in rare disease drug development, we need to consider the following factors comprehensively. One is the disease fundamentals, including the pathogenesis, the incidence rate, the affected population, clinical diagnostic markers, availability of suitable preclinical models, etc. These pieces of information can help us assess the difficulty of drug development and the size of the target patient population. Another important factor is whether there is any drug that has been approved for the specific disease. This may also include other information like the efficacy, side effects, the techniques, etc. From this, we can understand the demand for the drug to be developed and the scope for development. We also need to collect the market information like the risk and benefit assessment, pricing, the sales network, and investment situation, so on and so forth. In addition, the government policy is an important factor as well, such as the existence of expedited approval pathways, flexibility in clinical trials, availability of fund sponsorships, and finally, the collaboration across society, that is, whether the relevant diseases and patient populations have received attention from academia, industrial, governmental, and other sectors of society. The understanding on the disease and the drug accessibility are kind of fundamental, and the last three factors will further help drug developers to make initiation decisions. With advancements in science and technology, drug modalities have evolved from small molecules like inhibitor, modulator, to protein peptides like enzymes, cytokine, or antibodies, and then to nucleic acids such as oligo, microRNA, and then cell therapies like stem cells and CAR-T. This increasing complexity has enabled modulation of targets from phenotypic to genotypic levels, meeting the demand for more precise and effective interventions in diseases. Different modalities have been developed for rare diseases, and in some cases, there are multiple modalities approved for a specific rare disease. And I will delve into the current status and trend of different modalities for rare diseases. As we may know that approximately half of the approved drugs for rare diseases so far are small molecules. Here is a comparison between different modalities in terms of their mechanisms, molecular targets, target localization, and delivery methods. From this table, it is evident that small molecules possess comprehensive advantages. Firstly, the delivery methods are more flexible and diverse, accommodating various administration routes, including oral, injection, and inhalation. More importantly, they can act on all types of targets, including DNA, RNA, and proteins. For example, the gene tag molecules developed by Design Therapeutics can target the GAA-CTG repeat expansion region on DNA to act on a diverse array of diseases. On the other hand, as an effective SMN2 pre-mRNA modulator, Evrysdi can modulate alternative splicing of SMN2 mRNA, stabilizing the protein and benefits patients with SMA type 1, 2, or 3. On the protein level, Parakind, developed by Agios, is an allosteric activator targeting pyruvate protein kinase, which increase PK activity in PKD patients, alleviating red blood cell energy deficiency and chronic hemolysis caused by PKR gene mutations. Talking about protein as a therapy, which is also known as enzyme replacement therapy when applied in enzyme protein deficiency disorders, the most prominent indication for this therapy is lysosomal storage diseases, a group of metabolic rare diseases characterized by substrate accumulation due to lysosomal enzyme defects. However, this therapy faces numerous challenges such as high immunogenicity, poor tissue targeting, and short half-life. Recently approved for the treatment of Fabry disease, Elfabrio undergoes page modification of recombinant human GLA, significantly extending its half-life and reducing infusion frequency and dosage. Pombiliti is a unique recombinant human acid GAA enzyme with optimized carbohydrate structures designed to enhance uptake into muscle cells. Its combination with oral enzyme stabilizer helps stabilize the enzyme and minimize the loss of enzyme activity in the blood. These targeted strategies have actually turned the challenges into opportunities. Another important type of protein serving as a treatment is antibody. Monoclonal antibodies have been the most sought-after modality in the 20th century, and innovations in antibody engineering are offering the opportunity to design better antibodies with enhanced properties to maximize efficacy. For instance, antibody conjugates, glycosylation modified antibodies, antibody fragments, and bispecific antibodies demonstrate significant potential in rare disease treatments. For autoimmune rare diseases mediated by autoantibodies such as myasthenia gravis, a highly effective treatment strategy involves accelerating the clearance of autoantibodies by competitively binding to FcRn. Rystiggo and Vyvgart are two approved antibody drugs based on this mechanism, and AOC1001, on the other hand, is a promising antibody oligoconjugate for treating DM1, relying on the antibody portion targeting transferrin receptor 1 on muscle cells to successfully deliver siRNA, inhibiting the transcription of toxic DMPK within muscle cells for precision therapy. Finally, the bispecific antibody emicizumab, which can simultaneously bind to coagulation factors F9 and F10, mimicking the function of F8, has emerged as a promising option in treating hemophilia A with F8 defects, providing an alternative to ERT for hemophilia A patients. Moving on to oligonucleotides, we know that in recent years, many RNA therapeutics have been approved for the treatment of rare diseases. This is attributed to both the genetic mutation characteristics of rare diseases and the efficient and precise regulatory capabilities of oligonucleotides on DNA and RNA, enabling gene silencing, supplementation, and other functions. Here are 3 drugs approved in 2023 that utilize either siRNA or ASO to downregulate the expression of mutated genes for therapeutic purposes. Lastly, gene therapy. It is a therapeutic approach that involves the introduction, modification, or regulation of genes within an individual cells to treat or prevent disease. In vivo gene therapy and ex vivo gene therapy are 2 approaches used in this field to deliver therapeutic genes into target cells or tissues. In vivo gene therapy involves the direct administration of therapeutic genes into the body, where they are delivered to target cells or tissues. Therapeutic genes can be delivered using viral or non-viral vectors, while ex vivo gene therapy involves the isolation and genetic modification of cells outside the body before reintroducing them into the patient's body. Since over 70% of rare diseases are genetic disorders, gene therapy holds great promise for treating rare diseases. AAV, due to its higher safety, lower immunogenicity, and stable expression and tissue specificity, is currently the most successful commercialized in vivo gene therapy vector. By delivering different medicine products, it can achieve various in vivo therapeutic strategies, including gene replacement, modification, silencing, and transplanting. Among them, delivery cDNA for gene supplementation is the most approved strategy to date. Representative drugs such as Roctavian, Elevidys, Hemgenix can provide lifelong benefits with a single injection in clinical practice, and hence commanding high prices of course. In vitro hematopoietic stem cells gene therapy has been successfully applied in rare blood disorders such as sickle cell disease. Through ex vivo modification of autologous hematopoietic stem cells, correcting the mutated beta globin gene or inducing the expression of fetal gamma globin, and then reinfusing them back into the patient's body. Further differentiation leads to the production of red blood cells with stable hemoglobin structure and addressing the patient's anemia issues. I'd like to share case studies that with multiple modalities for the same indication. PKU is a rare disorder caused by mutations in the PAH gene, resulting in abnormal phenylalanine metabolism. The modalities involved in PKU treatments currently include small molecules, enzyme replacement therapy, mRNA, AAV-based gene therapy, and cell therapy. Among them, small molecules and ERT have approved drugs. We know Kuvan and Palynziq, while AAV and mRNA therapies are currently in clinical research stages, and cell therapies is only used in preclinical research. During drug development, different modalities need to assess whether PAH expression and activity are restored in vitro and in vivo models, and whether phenylalanine levels are effectively reduced in vivo. Different modalities require different techniques and methods to study their in vivo PK properties. For example, LCMS is often used to measure the in vivo exposure of small molecule drugs, while ELISA and MSD are used to quantify recombinant proteins in ERT therapy. For oligo and gene therapy, quantitative analysis using PCR techniques is essential. Additionally, due to increased complexity and molecular weight, immunotoxicity and off-target toxicity, among other factors, need to be assessed as important parameters for safety evaluation, in addition to routine toxicity testing. The second case study is about hemophilia A. With currently 6 different modalities in various stages of research and development, among them, enzyme and antibodies have marketed products. Similar to PKU, there are common features across different modalities in terms of PD and efficacy assessments, which involve evaluating coagulation factor expression, activity, and improvement in clotting ability. Regarding drug PK, due to differences in molecular forms, it is necessary to develop different analytical methods. Different modalities offer targeted approaches to address the underlying genetic or molecular abnormality associated with rare diseases, with advancements in gene therapy and mRNA therapeutics showing particular promise in clinical development. As a crucial foundation for drug development, appropriate rare disease models are particularly important. Researchers can utilize a variety of model systems ranging from simple in vitro cell lines to small animals and large animals to elucidate rare disease mechanisms, identify gene networks and therapeutic targets, and evaluate candidates. Each model system has its advantages and limitations, and the choice of model depends on a careful assessment of its translational relevance, accessibility, scalability, predictive values. Not only can these models aid in research and better understanding the underlying mechanisms of the disease, but they also serve as critical intermediaries for safety and efficacy evaluation, facilitating the advancement of drug clinical applications. Regarding the establishment of rare disease in vitro models, there are 2 main strategies. The first involves identifying mutations from patients by sequencing analysis and introducing these mutations into cell lines to construct stable cell lines. The second strategy is to isolate fibroblasts or blood cells from patients and reprogram them into iPS cells, and then induce their differentiation into specific cell types. These cells carrying patient mutations, after phenotypic validation and molecular mechanism studies, can serve as reliable in vitro models for high-throughput drug screening and are applicable for the development of different modalities. Compared to in vitro cell models, animal models are more reliable in validating genetic associations, understanding gene function, and developing therapeutic interventions. This is particularly important for rare diseases. Rodents are the most common type of mammal employed in experimental studies, and extensive research has been conducted using rats, mice, hamsters. Among these rodents, the majority of genetic studies, especially those involving disease, have employed mice. not only because their genomes are so similar to that of humans, but also because of their availability, ease of handling, high reproductive rates, and relatively low cost of use. Generally, rare disease animal models could be induced by genome modification, chemical or antibody injection, or surgical manipulation. Let's take a look at an example. It's also about PKU. So patients with the point mutations of PKU could be introduced to CAST1 cell line, and the expression and activity assay in vitro indicate that different mutations lead to the diversity of PAH protein stability and activity. And R408W exhibited the most severe phenotype in cell lines. And after successful construction of mice carrying this specific mutation, the lower body weight and lighter coat color, higher blood and urine PHE levels are found in the mutant mice, which resembles the features observed in PKU patients, indicating that this mouse model is suitable for drug development use. So the use of both in vitro and in vivo models accelerates the drug discovery and development process by providing valuable insights at different stages, from target identification to preclinical testing, thereby increasing the likelihood of success in clinical trials. I will stop here, and I would like to pass the floor to Dr. Lin Liu, and he will share his understanding and insights into rare disease drug development from the perspective of a senior drug developer. Dr. Liu, please.
Hello, everyone. So today I'm going to share some historical lessons, current status, and future expectations in the development of treatment for lysosomal storage disorders and other less rare diseases. So let's spend the next a few slides to discuss the background of the rare disease and lysosomal storage disorders. So the World Health Organization defines a rare disease with a prevalence of 6.5 or less in 10,000 people. It is estimated that there are more than 7,000 rare diseases, and around 72% of them are genetic diseases, which are caused by a single gene mutation. Lysosomal storage disorders is a group of rare disease of 50 different rare or ultra-rare genetic diseases, characterized by a specific lysosomal enzyme deficiency and resulting substrate accumulation. So the lysosome is a organelle, suborganelle, contains 50 or more lysosomal enzymes, which can break down macromolecules such as nucleic acid, proteins, and polysaccharides. Currently, there's no curative therapeutics for lysosomal storage disorders. So the approaches to treat lysosomal disorders are including enzyme replacement therapy, gene therapy, and small molecule therapy. So today I'm going to focus on the enzyme replacement therapy and gene therapy for this group of rare diseases. The enzyme replacement therapy has been developed in the last 60 years. So currently there are 16 enzyme replacement therapy has been approved by FDA to treat 10 different lysosomal storage disorders. So including Gaucher type 1 disease, Pompe disease, and Fabry disease. These diseases has more than 3 ERTs on the market. So the first ERT was approved by FDA in 1994 to treat type 1 disease. And more recently, there's 2 drugs approved for Pompe disease, which enhanced the muscle delivery to treat Pompe disease. So if we look back the past 60 years of the development of enzyme replacement therapy, so we can categorize to 3 stages. The first stage we call it the concept stage, and the second we discovery stage, and the third called development stage. So the most important stage I would consider is the discovery stage. So this stage takes around 2 decades. So in 1970s and 1980s. So due to in these 20 years, so the discovery of mannose 6-phosphate moiety and the mannose receptor and the mannose-6-phosphate receptors greatly facilitated the development of enzyme replacement therapy. Because during that time, so the whole field can understand the clear mechanism for this particular disease and how the drug can be delivered to cells. 3 people I would like to mention, let's say, during the field scientists. So in 1972, Dr. Elizabeth Newfield and Dr. William Sly labs confirmed the mannose-6-phosphate inhibits the uptake of beta-glucuronidase and alpha-arginidase in fibroblast cells. And the Km for the enzyme internalization is around 3 nanomolar, they calculated. So 3 years later, Dr. Stuart Cornfeld at Washington University Medical School calculated the structure of phosphorylated high mannose carbohydrate group in lysosomal enzymes and discovered the key enzyme in the generation of mannose-6-phosphate. 8 years later, his lab identified 2 mannose-6-phosphate receptors, cation-dependent mannose-6-phosphate and cation-independent mannose-6-phosphate, which can be utilized for enzyme internalization to all cell types. There are 3 key milestones for the success of the enzyme replacement therapy. First thing, understanding the cause of the disease. For example, the Gaucher type 1 disease is due to the abnormal macrophage, but the other cell types are not involved too much in the Gaucher type 1 disease. Second, discovery of enzyme delivery mechanism. Mannose receptors was discovered on the macrophage can be used for beta-glucocerebrosidase enzyme delivery to the Gaucher type 1 disease. Mannose-6-phosphate receptor pathway discovered can be utilized to deliver enzymes to all other cell types, including fibroblast cells or CNS cells. The third, development of the enzyme manufacturing system. The suspension chill cell system was discovered in more than 3 decades ago. So the enzyme by overexpressed in the chill cells has been found at the low levels of mannose-6-phosphate level. So these low levels of mannose-6-phosphate modification can be utilized for cell uptake. So the limitations for current enzyme replacement therapy in the field is poor mannose-6-phosphate level. So as shown here is the R-glucosidase alpha used to treat Pompe disease on the market. Only 27% of the enzyme has the ability to bind to the mannose-6-phosphate receptor. This indicates a core of the enzyme contains the mannose-6-phosphate moiety shown in the graph. And due to the large molecular weight of the lysosomal enzymes, they cannot cross blood-brain barrier. So the current enzyme replacement therapy drug under clinical trial or development are focused to overcome the limitations for the large molecule. So they either enhance the CNS delivery, fuse the enzyme with transferrin receptor antibodies or transferrin receptor binding domains. The second is people is also develop the enzyme for enhanced lysosomal targeting or cellular internalization, such as our company at M6P Therapeutics. So what we are doing now is that we developed a platform technology. This platform can be used in the producing cell lines, which can making the ERT enzymes. And we can also incorporate our platform to virus or non-viral gene for gene delivery. So our platform can ensure the enzyme produced in vitro and in vivo contains high levels of mannose-6-phosphate for cellular uptake. So as shown here is that the mannose-6-phosphate structures. So I would like to share a piece of the data for our Pompe project. So we have expressed the GAA enzyme in our platform designated as M021. So the enzymes was applied to the patient fibroblast cells for cell uptake experiments. So as you can see in the left, dark blue, the M021 has around nanomolar Km for enzyme uptake to the patient fibroblast cells. And the gray color showing the R-glucosidase alpha has much lower Km for cellular internalization. This indicated the moiety of mannose-6-phosphate important for the internalization of the enzyme for ERTs. So Pompe animals was also utilized to treat with M021 or the regular standard of care R-glucosidase alpha. So as you can see by by 3 months treatment at 20 milligram per kilogram dosage, M021 can fully recover the grip strength of the Pompe mass to wild-type level, but our glucosidase alpha cannot. So this is the ERT data showing the benefit of the mannose 6-phosphate content in the ERT treatment. So when we develop the gene therapy, what we should consider. So let's look at the principle of cell and gene therapy. So it's used the cells, virus, or non-viral particles as a vehicle to express or deliver a normal gene to the patient, so that the gene delivered can make the correct protein in patient to treat or cure a disease. The efficacy depends on the efficiency of the delivery of the functional protein made to right cellular location. So currently, FDA has approved 9 cell and gene therapies to treat 8 diseases. 4 are cell-based gene therapy, 5 are AAV-based gene therapy. No cell gene therapies approved for lysosomal storage disorders, but there are 8 cell therapies trials for 8 lysosomal storage disorders. One therapy was approved by EMA in 2020 to treat MLD disease. 2 of them are stopped. In the AAV gene therapy side, the Gaucher, Pompe, and Fabry gene for the gene therapy for Gaucher, Pompe, and Fabry disease has more than 3 clinical trials ongoing. For the others, we have either 1 or 2. So in total, there are 39 AAV gene therapy trials for 18 lysosomal storage disorders. 14 of the trials are either stopped or failed. The main reason for the failure of cell and gene therapy is insufficient efficacy. So the problem of the insufficient efficacy for gene therapy, I think, is similar to the ERTs as what we found. First, the limited tissue and cell transduction. Liver, heart, spleen are still the main organs for AAV transduction when we deliver systemically. Inconsistency of protein enzyme expression level between small animal and the patient. The third will be ineffective enzyme targeting to suborganelles such as lysosome. So here is the example I would like to show to the audience. So we treated the small animal mouse with the AAV9 directly to the CSF. So we're trying to understand how much of the AAV9 can distribute to the brain tissue. The whole brain slide was stained with the base scope to understand the messenger RNA levels. As you can see in the olfactory bulb cortex area, so we did see pretty good signal of the RNA signal of the RNA. After calculation, we see more than 40%. But in the middle brain or back of the brain, we only see a couple % of the cells are light up. So indicated low transduction efficiency in the middle and back brain. So this will be for us to consider how we should overcome the limitations of cell and gene therapies. So in the vehicle side, the new AAV serotype for higher transduction or broad tissue distribution will be needed. So that many companies are making the different tropism for CNS or other tissue delivery. Examples are shown here. In the cargo side, new technology enables high cross correction should be also considered as the lessons we learned from ERT. Increase the mannose 6-phosphate level of the enzyme produced in vivo will be very important for high cross correction. This is as what we are doing at M6V Therapeutics. So fusion protein with IGF-2 peptide or with the motive for binding of a transferrin receptor can be also considered alternate pathway for high cross correction. I would like to show one example of the program we are doing for mucopolysaccharidosis type 3B. So we incorporate the NAGLU gene alone to AAV, or we incorporate the NAGLU with our M6P platform to an AAV, so designated as M042. The in vitro co-culture work was utilized to prove the cross correction. So the Borden cells, 203 T cells, was used as a producer to transduce with AAV2. And the patient MPS 3B fibroblast cells were seated to insert 24 hours co-culture with acid conditioned medium for NAGLU and the activity, as shown in the middle bar graph. The NAGLU alone virus and M042 yield similar NAGLU activity in the conditioned medium. But when we look at the fibroblast cells, patient fibroblast cells, the M042 can definitely has much higher NAGLU internalization, which indicated the high mannose-6-phosphate can greatly increase the cross-correction in vitro cell system. So we also treated the MPS IIIB mice with AAV9, M042 or AAV9 NAGLU. Showing in the left graph, the brain tissue homogenized was utilized to assay how much of mannose-6-phosphate content by CMPR binding. So as you can see in the blue colors, the M042 mice has much higher CMPR has much higher CMPR binding compared with the gray color. Tissue enzyme activity for NAGLU was also assayed in the middle graph. So at the lower dose level, the NAGLU alone virus or M042 can reach similar to the carrier level NAGLU enzyme activity. But when we look at the HEXB level as a biomarker in the small animal, the M042 can reduce the HEXB activity close to the carrier level, but the NAGLU alone virus cannot. So take those data. So the future expectation for us in the cell and the gene therapy, we would like to expect it. Gene therapy has been successfully to treat a particular organ or tissue, such as eye, blood. A key question will be how to treat other large organs or tissues, such as heart or brain. Cell and ex vivo gene therapy, sufficient expression level, and the correct protein post-modification are important for a good efficacy. In vivo gene therapy, I think this would be ideal treatment for most of the disease. Vehicle side, AAV is the most common vehicle for gene delivery, but cannot redose. Nanoparticle for gene delivery would be a better choice. More research will be needed in the nanoparticle field. The new AAV serotype or nanoparticle for broader tissue distribution, higher transduction efficiency will also be needed in the field. So this could be reduce toxicity, cost, and increase efficacy for the treatment. In the cargo side, maintain sufficient gene therapy is very important. The protein or enzyme expressed cannot be too high or too low. Good post-modification with a protein made in vivo for correct subcellular localization should be also considered when we develop a in vivo gene therapy. Temporal or spatial gene therapy regulation will be also considered for certain diseases. So I would like to thank all the audience for your attention.
Thank you, John, for the presentation and insights. At WuXi AppTec, our goal is to enable you to accelerate your therapeutic programs so that we can get treatments to patients as fast as possible. For the discovery and preclinical phases, we provide end-to-end solutions from target to IND. We have a full range of capabilities, including molecular cell biology, hit finding, in silico bioinformatics, in vitro and in vivo pharmacology, through to DMPK toxicology and IND-enabling studies. I would also like to highlight that we are able to synthesize and produce small molecule, oligo, peptide, plasmids, and cell gene therapies at discovery scale, which we do not have time to discuss in this webinar, but please feel free to reach out to us for further information. Beyond IND, we have manufacturing and clinical services, supporting programs all the way to commercialization. Our expertise and experience covers a wide range of target classes, therapeutic areas, and modalities, so we can support you for a wide range of therapeutic strategies to create new therapies for rare disease patients. Thank you very much for listening.
Thank you all once again for tuning in today, and thank you to our speakers for their superb presentations. In a few moments, we'll move into the live Q&A session, so please submit your questions if you haven't done so already. Before we start the Q&A, a reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please keep an eye on your email and do share the link with your colleagues who might not have been able to tune in today. All right, just give us a few moments to transition to the Q&A session. Hello, everyone, and welcome to the live portion of this webinar. Thank you so much for joining us. Thank you for all the questions that you've been sending in. All of our speakers are here and they are ready to take your questions. So just a reminder, if you haven't done so already, if you do have a question, feel free to enter it in the Ask a Question panel on your screen. We're going to be taking questions live for the next several minutes. All right, I'm going to jump right ahead into the questions because we do have quite a few to get to. First question I have, do you think enzyme replacement and substrate elimination combination therapy is a better option for enzyme deficiencies like LSD? Lynn, I think this one's for you.
Okay, thanks for the question. So I would agree with that for certain LSDs. So I did see certain companies that develop, like say, a substrate reduction. So the strategy, what they are doing is that, so they take the ERT the first, like say, maybe a few months. So like say, because the ERT can further benefit in terms of reduce the substrate in the beginning. So then they only switch to the small molecular in the substrate reduction side to maintain the levels. So that could be a good choice. I would agree. If the only thing is that, so if the substrate reduction, the small molecule can function well, so that means you control the certain level, not like say to clear the substrate to too low, because you need a certain lipid or glucose or glycogen in the cell for certain function.
Excellent. Thank you so much for that. All right, next question I have. When assessing the strengths and weaknesses of non-viral versus viral vectors in gene therapy, do you think that future research trends will focus more on one of these approaches over the other? Joanne, can I come to you with that one?
Yeah, sure. I'll take that question. So, from my knowledge and understanding, the future of gene therapy vectors is likely to involve both viral and non-viral vectors because each of them has its own advantages and applications. For instance, the viral, especially for AAV, has the nature of highly efficient gene delivery, long-term gene expression, and can target specific tissue or cell types. And these features are kind of unreplaceable, at least for now. And these features make them in application where precise targeting and stable change gene expression are critical. And on the other hand, the non-viral vectors hold good safety and large cargo capability, as Dr. Liu just shared, and they're easy to be manufactured. So, and similarly, these are also crucial in clinical use, making them attractive candidates for certain gene therapy applications, particularly those requiring large cargo capability. So I think the future of gene therapy is likely to involve a combination probably of both viral and non-viral, with each vector type offering unique advantages and addressing specific challenges. I hope I have answered the question.
Yeah, absolutely. Absolutely. Thank you. Next question I have, does improved cell uptake work on only specific cell types? Who'd like to jump in on this one?
So I would say it depends, right? So it depends on which pathway you use. So let's say take what we are doing now at our company, M6B Therapeutics, we use the mannose-6-phosphate receptor pathway. So if you trigger, if your molecule can target to this particular pathway, so I believe the enzyme or the other large molecule can target to almost every cell type. So based on the current findings, that so the mannose-6-phosphate receptor are identified in all the cell surface, all the cell types. But if you use the other serotype, the other receptor for large molecular targeting, that could be to only specific cell types, right? So for example, the transferrin receptor right now, most people use for blood-brain barrier. So a concern will be how much will be that involved in certain cellular uptake into certain cell types.
Excellent.
So yeah.
Yeah. Thank you. Thank you. And just to remind our audience, we're taking questions live. So if you do have a question, please go ahead and enter it in the Ask a Question panel on your screen. Our speakers are answering questions now. All right, the next question I have, what is the duration of AAV expression in vivo? Joanne or Alice, would you like to take a stab at that one? Or anyone can jump in on it.
Maybe I can jump in, and the rest of people the team can follow. So for AAV expression, so the duration, that's a good question. I think that's also that's also like say all the scientists or the people in the field are trying to understand. So for the systemic delivery, because we know for systemic, the majority of AAV are targeted to liver, and normally the liver cells can, let's say, can expand quite a lot. That means, can divide a lot, during the growth, especially when you're AAV treated in the young age, right? So in this case, so we did see the expression level or the AAV in the liver is going to dilute it. So from our, let's say, small animal study, we injected those animals at adult age. And once we keep tracking for a year later, so we did see the whole expression level or gene messenger RNA level goes to below to 50%. So that's for small animal, just for 1 year. So I believe in the human, in the large animal, we see similar things. So, but what I heard around is that depends on the efficacy, how much of the enzyme you want to generate. So if, like say, 10 years later is still above the threshold, can introduce a certain efficacy, that could be okay. So I hope it's more than 10 years, but I think we need to wait and see more clinical data come out.
Excellent. Excellent. Thank you. All right, next question I have. What is the role of AAV9? And anyone can jump in.
Okay. So I can help a little bit. So what's the role of AAV9? So AAV9 actually is an engineered version of a serotype. So it binds to so what happens is that it introduced a certain mutation in the surface of the capsid protein, so it can interact or bind to the surface called salicylic acid. So normally, the cell surface either the liquid, either the lipid or protein, you have a certain sugar modification. So you're exposed to terminal salicylic acid. So the AAV9 can bind, interact with salicylic acid. So that could be internalized through the membrane, let's say internalization or endocytosis. So I think that's then the AAV9, because once it's capsulated to the vesicle, be internalized to the cell, then it can be targeted to endosome lysosome and escape from the endolysosomal system and deliver to the nucleus for expression. I hope that I answered the question correctly, because the role of AAV9, I believe it's kind of similar as the other AAV, 1 or 2, 8 or 10 of the others. I can more consider this AAV9 or AAVs as a vehicle. So as let's say a cargo, you package those cargo inside. The function part is still the cargo.
Excellent. Excellent. Thank you. Next question I have. Long term, how would nanoparticle gene therapy improve upon AAV efficacy, excuse me?
I think it's again Dr. Liu's expertise.
Sure.
Oh, okay. So this is good point. So my thoughts is that, so I think the nanoparticle gene therapy that's a very have a unique benefit there. So for example, let's say take the AAV gene therapy. If people, like the patients involved in the AAV gene therapy, you need to do the screening of the neutralizing antibody in the blood. So based on the current data suggests overall, there's around 30 to 60% of the people has to be excluded from the treatment because they have a high level of neutralizing antibody. Then the question will be how you're going to treat the others, right? So the nanoparticle, let's say gene therapy, the benefit is that there's no consideration of the neutralizing antibody, and you can dose every single patient, and you can redose. What's the point is that, so let's say for certain diseases, depends on the efficacy point. The 1 year or 2 years later, after the one dose of nanoparticle gene therapy, if you need it, if the patient need it, right? Because every patient behaves differently in terms of the expression and the response to the treatment. So you may need 1 year later another dose, but AAV gene therapy cannot. After one dose injection, you cannot do another one. So in terms of, for this point of view, so for the nanoparticle gene therapy, I don't think that would be consideration for how long the long term. I don't think that would be a consideration because you can redose, right? But for AAV gene therapy, because it's a one-time work, one-time thing, so you have to think about how long it can last. So I hope I answered the question. Yeah, thanks.
Yeah, of course. Thank you. That's great. And just to remind our audience, we're taking questions, so we have time for a few more. If you do have a question, please go ahead and enter it in the Ask a Question panel. All right, the next question I have, the construction of animal models for rare disease usually takes a long time. Will this hinder the development of drugs for related indications?
I'll take that question.
Yes, please.
Yeah, thank you for this question. It's a good one. And my short answer is yes. The construction of animal models for rare diseases can indeed be time-consuming and sometimes even challenging. And this process may potentially hinder the development of drugs for related indications. And in some cases, especially for CNS-related rare diseases, it is difficult to get a model that is good enough to mimic the symptoms of patients due to their unclear and complicated pathogenesis. And this process can be lengthy and may delay the initiation of preclinical studies and the subsequent drug development activities. But I think there are some strategies that potentially can help mitigate these obstacles. For instance, the collaborations among academia and industry partners and even patient groups can facilitate the sharing of resources and expertise or even data to accelerate this model development process. And on the other hand, in some cases, in vitro models, for instance, iPS cells and organoid systems might be a good supplementation or even replacement. Yeah, I hope I have answered the question.
Yeah, absolutely. Thank you so much for that. Next question I have, drug repurposing or indication expansion is an important approach for rare disease drug development. Do you think it's still necessary to perform preclinical studies before clinical trials?
I can take that one.
Sure.
Thanks for this good question. Yes, even in cases of drug repurposing or indication expansion for rare diseases, it's typically necessary to conduct preclinical studies before advancing into clinical trials. While the drug may have already been approved for another indication, but the specific disease mechanisms and patient population and also potential safety concerns associated with the rare diseases indication may differ from those of the original indication. So conducting preclinical studies before clinical trials is sometimes essential for ensuring the safety and efficacy of the repurposed drug or indication expansion. in the context of rare diseases, and for meeting the regulatory requirements.
Excellent. Thank you so much. We have time for one last question from our audience. So the question is, are there any organ-on-chip-based systems used in screening candidates in the rare diseases drug discovery space? And second part, can you elaborate on the use of these in drug discovery? And Lynn, this one's for you.
Okay, this is a good one. So I did see that I would say yes. So for the especially for the chip side, I did see there's a companies still starting to develop some kind of chip you can do the screening. So and it can and also certain companies so especially for those brain tissue. So they have similar organs they're trying to culture inside to mimic the disease model or function. I believe this is a good strategy can do. But the only concern is that so I think this is also very good for in vitro, especially when you're screening your drug and you want, let's say, quick assay, and we want to understand how your drug behaviors. That would be, I would think this is good tools to do, and it's very good for the discovery work. I believe for that. So what's the second question?
Can you elaborate on the use of organ-on-chip-based systems in drug discovery?
Okay. So for the use, I think most of people currently use for screening. So but for the animal model side, especially for the efficacy evaluation, I would at least for my view, I haven't seen a very good model or way to do that. So, but I would love to see any new technology come out and to work with the rare disease field and together with us and try to solve the problem in the field. So, thanks.
Excellent. Excellent. Wonderful. Thank you all so much. That is all the time we have for today's webinar. For our audience, as a reminder, this webinar will be available to view on demand in the next 24 to 48 hours. So please keep an eye on your email. You should get a link in that time frame, and you can review the entire webinar, including the Q&A, and also share the link with your colleagues who might not have been able to join us today. I'd like to thank all our speakers. So thank you, Lynn, Joanne, and Alice for being here for your presentation and for taking our questions. A special thank you to today's sponsor, WuXi AppTec, for sponsoring this webinar. And I would also like to shout out the Gen Multimedia Group for hosting. So thank you, Hannah, Liana, and David. And last but not least, excuse me, thank you to the audience. Thank you for being here and for asking great questions. From all of us at Gen, thank you so much and have a wonderful day.